In Chapter 4, we explore our first departure from Mendelian inheritance with the concept of sex and sex linkage. In Chapter 2, we discussed how meiosis produces haploid gametes. Those haploid gametes then undergo fertilization to become a diploid adult. This is the process of sexual reproduction. Sexual reproduction has evolved many times in eukaryotes, and in many of those instances we have males and females. Although there are still many asexual species, particularly in protozoa, sexual phenotypes, like male and female, have many benefits. These benefits include being specialized to fight for access to resources and to take care of and produce young. In some species, these sexual phenotypes are controlled by sex chromosomes. These originated as autosomes, but then differentiated into having genes that control the sexual phenotype. Since they originated as autosomes, many sex chromosomes still have autosomal genes. In this depiction, each blue tick on the chromosome is a locus where a specific gene is. This introduces us to the concept of sex linkage. Some autosomal genes that control a phenotype that has nothing to do with sex will be linked to the sexual phenotype because they're on the same chromosome. It's important to note that sex has evolved and disappeared many times throughout eukaryotes. There are many ways that sexual phenotypes can be determined, most of which are genetic, but some are environmental. For example, some reptiles lay their eggs in the ground where the ambient temperature is relatively constant. The eggs near the top are going to be slightly warmer and develop as one sex. The eggs near the bottom are going to be slightly cooler and develop as the other sex. It happens both ways in different species. With respect to chromosomal sex determination, mammals have XY sex determination, where the SRY gene is on the Y chromosome. If you have the Y chromosome, you develop as male. If you don't have a Y chromosome, you develop as a female. Some species have XO sex determination, where there's only an X chromosome, and it's the number of X chromosomes that you have that determine if you're male or female. There's a certain threshold beyond which you develop as either male or female. It happens both ways in different species. In XA sex determination, it's the ratio of your X chromosomes to your autosomes that determines your sexual phenotype. This is the case in fruit flies and many other insects. If your XA ratio is 1/2 or lower, you're male. If it's 1:1 or higher, then you're female. In chromosomal systems like in mammals, females are XX, they're homogametic, and males are XY, they're heterogametic. But sex determination has evolved using different chromosomes in different species. For example, birds use Z and W chromosomes. In birds, ZW is the female, heterogametic, and ZZ is the male, homogametic. The homogametic versus heterogametic sex becomes very important when we start to discuss the consequences of sexual phenotype on Mendelian inheritance. As we discussed in Chapter 2, in meiosis, two autosomes, homologous chromosomes, line up with each other during crossing over of prophase. If we're discussing the homogametic sex, like the female in mammals, then this isn't an issue because she has two X's and they can line up with each other in prophase. The process of synapsis in prophase is determined by similarities in the base pairs in the homologous chromosomes. Because the two autosomes are nearly the same and the two homologous sex chromosomes are nearly the same, they can line up next to each other. This represents a problem for the heterogametic sex because they have two different chromosomes, so they don't have any natural way to line up with each other. The X and the Y chromosome in male mammals cannot find each other in prophase. They solve this problem with what we call the pseudo-autosomal regions, At the end of every sex chromosome, there is a pseudo-autosomal region that is the same across the chromosomes. So X and Y chromosomes can find each other in prophase thanks to the pseudo-autosomal regions. The applied content of Chapter 4 deals with the consequences of sexual phenotype and sex linkage from Mendelian inheritance. In Chapter 3, we introduced the concept of a Punnett square, and we talked about how you can take two diploid individuals and predict the phenotypic ratios in their offspring using a Punnett square. However, this basic setup only works if we are discussing autosomal chromosomes and autosomal genes. Whenever we're working with sex-linked traits or genes that are on a sex chromosome, we have to use superscripts. In this example, I've drawn my X chromosome, and then with my superscript, I've indicated what allele that chromosome has. So my chromosome either has a big A allele for yellow pea color or a little a allele for green pea color. Note that I have a Y chromosome that doesn't have a superscript. That's because my Y chromosome does not have the trait in question. Whenever we're dealing with species where males are the heterogametic sex, then that male will always be hemizygous. He's only going to have one copy of all X-linked traits. After some slight differences in my setup, my process is now the same. I fill in my Punnett square just the same as I would if I was talking about an autosomal trait. But because things get a little bit more messy, I'm going to pull all my values from the boxes of the Punnett square off to the side. Now I can look at each individual value and determine its phenotype. For autosomal traits, there's only one trait that I'm interested in, especially for a monohybrid cross. But for sex-linked traits, even for a monohybrid cross, I have two phenotypes to keep track of. I need to know if the individual is male or female, and I need to know what color pea they produced, either yellow or green. All my XX individuals would be female, and XY would be male. And as long as you have one copy of the big A, then you're yellow, but if you don't, then you're green. In this example, my ratios are 1:1 for both males and females. Oftentimes, when we're dealing with sex-linked traits, I will see different ratios in my male offspring and my female offspring.